All right, so we are Bio 208. This is fall 2020, and we are in week 2 of our class, and we are starting the physiology of the heart, which is chapter 20 material. So, the part of heart muscle tissue. So, if we go back to chapter 4, we learned that there are 3 types of muscle tissue. We learned there's smooth muscle, there's skeletal muscle, and there's cardiac muscle. And we learned that on a spectrum, smooth muscle is on one side of the spectrum and skeletal muscle is on the other, and that cardiac fell in the middle. Okay? And we learned so far that cardiac cells are single cells. They have a distinct kind of entity to them. They are not long and skinny, and they're not spherical. And sometimes because of the connective tissue, they might have places where the cell kind of forks or breaks or kind of has make space for connective tissue and blood vessels, especially capillaries, to be able to push through and provide oxygen and fat and protein, glucose for ATP production, and to take away their carbon dioxide and their waste, their hydrogen ions. Okay, so that's what we know about cardiac tissue. We know it's striated. So we know there's going to be sarcomeres. We know that that means actin and myosin have to interact. And because the sarcomere is going to have troponin and tropomyosin, we know that calcium is going to have to bind to troponin to be able to pull tropomyosin off. And I'm working. Can I? Over there. And we know that calcium is going to be in a sarcoplasmic reticulum. All right, and so the steps of what happens to get a cardiac cell to contract are going to be somewhat related to how we got a skeletal muscle to contract, and we learned about that in chapter 10 last semester. Now, there are some differences between skeletal and cardiac muscle, all right, and one of the big differences is that there is no neuromuscular junction. There's no neuron telling a cardiac muscle to contract. So all of the cardiac muscles in our heart are going to be told to contract by a different mechanism. And that mechanism is going to be that some of our cardiomyocytes are going to differentiate into pacemaking nodal, N-O-D-A-L, like I have written up here, these nodal cells. And they are going to, in some ways, take the place of the neuron that's telling the rest of the cardiac muscles when to contract. Okay, so one of the big differences between the cardiac cells and the skeletal muscle is there's no neuron telling them when to contract. So there's no acetylcholine release, there's no acetylcholine receptors, there's no acetylcholine esterase, there's no resting membrane potential of these cells that is sitting at a flat negative 80 millivolts or at negative 90 millivolts waiting for acetylcholine to bind to the receptor and bring sodium in. Okay, that's not the way the structure is going to work. All right, so because we have these different kind of nodal cell populations, they are setting the rate intrinsically. So without the nervous system telling them to, they are going to set the rate at which the rest of the cardiac cells are going to contract. Now, another big difference between the cardiac and the skeletal muscle is cardiac muscle cells are going to be touching each other, and they are going to, in some cases, bind their membranes with special proteins that are going to let the cytoplasm of one cardiac cell quickly flow into the cytoplasm of another cardiac cell. So they are physically and chemically linked up to each other. All right. So when these nodal cells trigger changes electrically that lead to calcium release from a sarcoplasmic reticulum, they are going to then be able to trigger not just the cells immediately around them, but all the cardiac cells, because all the cardiac cells are physically and chemically linked to each other. Okay? Now, the nodal cells intrinsically are going to have what's known as an unstable resting membrane potential. So that means that there are going to be molecules, ions, moving into and out of the cell. They're leaking in and leaking out. That is going to make the resting membrane potential flat lines like we see with our neurons, like we see with our skeletal muscle, and like we see with our contractile or our non-nodal cardiomyocytes. So if you look at my picture over here, this line here represents the resting membrane potential of most of my cardiomyocytes, most of my heart cells. Okay, and if I go to a slide down here, this is potential up here of the skeletal muscle cell. And you can see it is a flat line. It is not moving. All right. So most of the time, our resting membrane potential of cells is a flat line. So there's a balance between what is leaking in, what is leaking out, what is getting pumped, and that is a steady state environment. And so when we measure the electrical potential of that environment, we usually see a stable line. Okay, and in the neurons, that stable line is sometimes at -70, sometimes at -75, sometimes at -80. All right, because it's under the influence typically of the neuron's sodium, because sodium is usually going to come in, and it's usually under the influence of potassium, because potassium is hoarded inside of the cell because of the DNA and the RNA and the phosphate and proteins that tend to, in water, let go of a hydrogen ion and thus have a negative charge. So to keep things electrically, neutral, potassium as a positive molecule is the hoarded molecule. Okay, and so there's a lot of potassium inside the cell, not a lot outside. So potassium tends to want to float out, sodium tends to want to float in. We have pumps to reset, and so typically most cells, the resting stable membrane is a function of some sodium going in, some potassium going out, and an ATPase resetting the system. Okay? And that's what the video that I asked you to watch on the nervous system was about. What is the resting membrane potential, and what are the ions that usually control that? Okay, and usually what we then use as our link to then talk about skeletal muscle, smooth muscle, and most of the cardiomyocytes is their resting stable membrane potential is a flat line too, and their resting membrane potential, if it's not at -90, meaning nothing's moving, there's usually, again, a little bit of sodium leaking in, a little bit of potassium leaking out, and a pump to kind of reset things. And that's why the resting membrane potential might be at like -85, -80, -75, -70. Okay. And the more sodium leaking in, the more potassium leaking out, that usually means, again, the less negative your membrane potential. Okay. Nodal cells are not going to follow the general rule of what is happening in the neuron and with other cells for the resting membrane potential. Okay, they are going to have an unstable membrane potential. So the line, as it's shown here, is not ever flat. And again, when we go to the next slide, the line is not flat. So why is the line not flat? There is an unequal balance of molecules coming in versus molecules coming out, and there's not an ATPase helping keep the balance. All right, so the pacemaker, the depolarization, all right, is going to be that normally in our nodal cells, we are going to see sodium ions floating into the cell at a faster rate than potassium ions are floating out or leaking out. So more positive molecules are coming in than positive molecules going out. And so the amount of positive molecules inside the cell is then becoming higher. And so the value of what the millivolts is on the voltage meter is becoming less negative. Okay, so the pacemaker potential, the slow depolarization potential of a nodal cell is going to be a slope on this line. And that slope is going to be, again, determined by the rate of sodium coming in and usually the rate of potassium going out. The faster or the more permeable we make the membrane of the non-contracting, non-resting cell to sodium, the slope of the line is going to become more straighter. So we are going to reach those threshold voltages faster. The sodium being less permeable or slowed down or blocked is going to make the slope of the line flatten out, and it's going to take longer to reach threshold. All right. So while cardiac nodal cells are intrinsically going to have sodium leaking in versus potassium leaking out and are going to have an unstable resting membrane potential, that's going to set the rate for the heart and how many times it beats. That resting membrane potential, that pacemaker potential, that slow depolarization can be influenced by the nervous system, by local factors, by drugs, by hormones, and we can manipulate that slope of sodium coming in versus potassium and thus the slope of the pacemaker potential to be reach threshold faster or reach threshold slower, and that is going to be how we influence heart rate. Right? Now, if we were to cut open our bodies and take our hearts out, intrinsically with the heart not getting any influence from the nervous system, not getting any influence from hormones, not getting any influence from any molecules or drugs, your heart should beat about 88 times a minute. So that means the rate that, again, sodium typically flows in versus potassium flows out is going to have a slope of the line that's going to generate contraction and action potentials on the entire muscle of the heart about 88 times a minute. If most of you who are wearing a Fitbit look at your heart rate, mine's at 78, obviously I have influences on the slope of my pacemaker potential line that's causing it to be slightly flatter, so therefore the number of times my heart beats is less than that 88 times a minute. Okay? So your takeaway. Nodal cells have an unstable resting membrane potential. That resting membrane potential is an influence of sodium leaking in, potassium leaking out, and they're not going to ever balance it, so it's constantly moving towards threshold. All right? The slope of the line, if we don't do anything and have no influence, the heart should beat 88 times a minute. All right? But we do influence that with the line, and when we do influence it, again, we can make the permeability of sodium higher, making the slope of the line reach threshold faster, making ourselves have the ability to contract more than 88 times a minute. Or we can try to block sodium from going in, increase potassium leaving. Again, all of that's going to make, again, it harder to reach threshold because the positives are not coming in as quickly and the positives are leaving more quickly. And that's going to help us, again, maybe slow down from 88 beats to less. Okay? So, questions to ask. What is the resting membrane potential? What are the ions that influence the resting membrane potential of a nodal cell? The answers would be sodium ions leaking in, potassium ions leaking out. And the rate that sodium is coming in, the rate of permeability to sodium versus the rate of permeability for potassium to leave. Okay, if I ask you what is the resting membrane potential of a contractile cell, it's negative 90 millivolts. Nothing's getting in, nothing's getting out. Okay, those cells are sitting and waiting to be told to contract, to be told to let their membrane-bound channels open because the voltage changes on their membrane because they are physically and chemically linked to these nodal cells. Okay. Now, the action potential part. Once you reach threshold, this critical line on the membrane of the cell. So remember, your cell, has biphosphate, membrane. there are proteins in the cell that have closed doors. Those proteins, when the voltage on the cell reaches critical value, those doors are going to open. And when they open on a critical voltage, they are known as voltage-gated channels. For the nodal cell, the rate of rapid depolarization, meaning the rate of change of the membrane voltage across it, going from a negative value to a more positive value, that's the rapid depolarization. It's sometimes known as phase zero, all right? That rate of rapid depolarization is going to be because of calcium ions coming into the cell. That's new. For the action potential in the previous cells that we've talked about, the nervous cell and the skeletal muscle cell, depolarization is almost always a sodium event. So this is a new point where we see rapid depolarization in the nodal cell. So the action potential, the rapid depolarization, phase zero, is because not sodium ion, but calcium ions, calcium channels are opening, letting calcium into the cell. Okay? Now, skeletal muscles, we wanted calcium to be involved in the contraction, but all of the calcium we needed in a skeletal muscle is in the sarcoplasmic reticulum. So remember that sodium coming in, when acetylcholine bound to its receptor opens a sodium channel, a ligand-gated sodium channel, sodium coming in, that triggered depolarization on the membrane. That depolarization triggered the sarcoplasmic reticulums to open calcium channels, letting all the calcium that was housed in the sarcoplasmic reticulum to float into the cytoplasm and bind to troponin. Okay, in the neuron, the action potential doesn't really have a calcium role. It's sodium comes in, potassium goes out, and that travels down the membrane of the neuron until you get to the terminal part, and then you work on getting vesicles released. All right. So the action potential of a nodal cell, the rapid depolarization, this is the first time we've seen it. The action potential's rapid depolarization is because in the nodal cell, calcium ions are going to come into the cell at a rapid rate. Now, nodal cells still have sarcoplasmic reticulum and still have troponin. So the calcium coming in is going to directly open calcium channels on the sarcoplasmic reticulum. So it's calcium-induced calcium release, and more calcium is going to exit out of the sarcoplasmic reticulum. And the calcium that came in and the calcium released from the sarcoplasmic reticulum together are all going to go bind to troponin, and we're going to then get tropomyosin pulled off, actin and myosin hook in, and power strokes and cross bridges, and we're going to keep doing that as long as ATP is available. Okay? So calcium is going to play from outside of the cell coming in and rapid depolarization, and then once it's in the cell, because it's a muscle cell, it's going to then play a role in getting the calcium released from the sarcoplasmic reticulum, and then it's going to go bind to troponin and do what calcium does in a skeletal muscle cell, let actin and myosin interact. All right. Calcium channels are going to close. Calcium is going to start being pumped out. Calcium is going to start getting pumped back into the sarcoplasmic reticulum, but we need the membrane potential to reset. The repolarization, the hyperpolarization of the membrane is going to be just like it was in skeletal muscle, just like it was in neurotissue. The return to resting is going to be because potassium ions, and remember, potassium ions are the positive ion hoarded inside of cells. They have the high concentration inside the cell, the low concentration outside the cell. So when I need to get rid of a positive quickly, they are the molecule that I can quickly open channels and let flow down their concentration gradient. So they are going to be what flows out and reestablishes and repolarizes my nodal cell at the end as I'm ending my action potential and resetting. Okay? So, nodal cells, not a stable resting membrane potential. That slope of the line is because sodium can float in, potassium floats out. Typically, different things influence the rate of sodium permeability and potassium permeability. So the line is influenced by lots of things, but if nothing is influencing it, there should be about 88 resting membrane potentials triggering action potentials of the nodal cells. When I trigger threshold and I trigger on my nodal cell my voltage-gated channels to create an action potential, the first phase of an action potential is rapid depolarization, and for the first time I'm going to see rapid depolarization in a nodal cell because of calcium ions coming in. My repolarization will be like everywhere else because potassium flows out. And then I will start again to see sodium and potassium influencing the resting, getting me ready potentially with a tight line or a flatter line to get me to my next trigger, my next threshold to trigger my next heart contraction. Okay. Now, action potentials of the contractile cells. Their resting membrane potential is the flat line. So nothing is happening. And then these guys hit threshold. These guys have an action potential. These guys are physically touching some of these contractile cells. What happens? When these guys hit thresholds and these guys start seeing calcium come in and triggering rapid depolarization in their membrane, some of that energy is going to jump to my contractile cells. And that energy and that change in membrane voltage is going to, on my cardiac contractile cells in the atria and in the ventricle, going to trigger again a voltage-gated channel and a large number of them to open. And I'm going to see rapid, so many voltage-gated channels opening and so many positive ions coming in. This is almost an instantaneous straight line up. Okay, so within a few milliseconds, boom, I went from not having a lot of positive ions in my cell to a crap load. All right, this phase 0 of my contractile cardiomyocyte is sodium coming into the cell. So my action potential of a contractile cell, rapid depolarization, phase 0, is back to, like in the skeletal muscle, like in my neuro muscle or neuro tissue, is going to be sodium ions coming into the cell on a crap load of sodium ion voltage-gated proteins opening. Okay. Those voltage-gated sodium channels, again, when they hit a certain value of membrane potential, they are going to inactivate and they are going to close their doors and no more sodium is going to come in. Potassium is going to realize that the membrane potential is all screwed up. It's going to open voltage-gated potassium channels. And so initially I'm going to see this start to drop, and that's phase 2 or phase 1, I think. I can't read the numbers. Where, again, potassium, like over here, potassium starts to come into, out of the cell and starts to try to get things back toward normal. New piece. In a contractile cell, before I just let the line slope all the way back down below threshold and reset, I hit this plateau. This plateau represents potassium is still trying to go out of the cell and trying to repolarize and get things back to resting. But as I have a positive ion going out, I am going to have calcium coming in. Okay? So my cardiomyocytes need some calcium to enter the cell to trigger calcium release from the sarcoplasmic reticulum. Okay, so unlike skeletal muscles, there's no little triad, there's no T-tubules, there's no connection directly of the membrane and the sarcoplasmic reticulum to get calcium released from all of my sarcoplasmic reticulums. So I need a different mechanism. Second, in cardiac tissue, only about 80% of the calcium I need is inside the sarcoplasmic reticulum. So that means I need more calcium from another source because I don't have all the calcium I need for all the troponin binding. So I need in all of my cardiomyocytes a point in time where calcium comes in. So in my nodal cells, the calcium comes in for rapid depolarization phase of my action potential. In my nodal cells, calcium hasn't come in yet. It's because it's not coming in at rest. It's not coming in the rapid depolarization. So this plateau is a time where I get calcium in as I'm letting potassium out. So I flatline a little bit my action potential in a depolarized state. But I get calcium in. And again, I just need about 20% of the calcium. And I need some of those calciums to come in, trigger calcium release from my sarcoplasmic reticulum. Now I flood my cytoplasm with calcium to all those troponin molecules. And now when I contract, I'm contracting with more actin and myosin interactions than if I just depended on the calcium in the sarcoplasmic reticulum, or if I just depended on the calcium to come in from the membrane, which either way is not sufficient. I need both of those calcium populations together to get all of the actin and myosin interactions to generate an effective contraction. Okay, so that phase 2 of an cardiomyocyte contractile cell action potential is known as the plateau, and it represents the point in time where the calcium ions that I have to have come in to trigger calcium release, the calcium ions I need to supplement the 80% of calcium I have available in my sarcoplasmic reticulum, but I don't have enough total to get the contraction I want, come in and give me that supplementation. That's where that's happening. And eventually those calcium channels will close, potassium will continue to flow out, and so eventually, like over here, phase 3 will be the repolarization all the way back down to resting because potassium is flowing out and no more positives coming in to balance that out. Okay? The calcium that I bring in and the sodium that I bring in, eventually I will pump some of those molecules out with ATPases and calcium and sodium exchangers. And again, you'll learn more about that when you get to pharmacology because all of those proteins are going to be drug targets. All right. But I'm not going to see any more calcium and sodium coming in my repolarization. So that's why I'm going to see the action potential eventually go back to resting and reset. Okay. So that was a lot of information. You need to know for these cells, what is happening with resting conditions? What are the ions moving or not moving? When an action potential happens, what are the ions that are going to be triggering the rapid polarization? What are the ions that are going to be triggering for the contractile cells, the plateau? And then for both, what are the ions, again, potassium, that are going to be triggering the repolarization? Okay. This is all happening by voltage-gated mechanisms. So when I ask a nodal cell to, again, and that's the another important thing about this, remember, action potentials are repeatable, identical events. So when you ask an action potential in a neuron to happen, and then you wait and you let it reset and you ask it to do it again, the action potential looks exactly alike. Why? Because it's all voltage-gated channels letting things move until it hits a certain limit, and then it stops, and then it resets. So the action potential here on the nodal cell is always going to look alike. Right? And the reason why is because these are voltage-gated channels. What changes and what's stimulated are these pacemaker potentials. Okay? Same thing with cardiomyocyte cells. The action potential is always going to look like this. In skeletal muscle cells, it always looks like that. In neurons, it kind of always looks like that. Now, the importance of why is there this long in time action potential when normally in the neuron, in the skeletal muscle, and other places, it takes like milliseconds total. Okay? This long refractory period is going to be the reason why cardiomyocytes cannot have tetany, cannot have TREP, cannot have wave summation. So if you don't remember what those things are, go back and read chapter 10. And how did I get a skeletal muscle cell to give me more contraction when I asked it to contract again and again and again and again? How did I get the force that it produced to get bigger and bigger when the cell still had the same number of actin and myosin proteins? Well, it got bigger because frequency of stimulation, I would start to tell it to contract before it was done with the previous contraction. In the heart, I will not be able to do that because the action potential is such a long refractory period. Okay, and this is always a concept that people have a hard time with. Why would I not want the heart to have tetany? Why would I not want the heart to have TREP? What good is it for me to contract the heart with more force if I didn't give it time to relax and fill with blood? So why do I never want the heart to have TREP, to have tetany? It does me no good to make the heart contract a second time more powerful and more forcefully if I didn't give the heart time to fill with blood. Okay? So because of the action potential of the contractile cells, I am not going to be able to tell the heart to contract with more force by manipulating the frequency of contraction. And the advantage to that is then I can never make the heart try to contract before it's had a chance to fill with blood. Okay? Now, can I make the heart cell contract more forcefully with the 1,000 actin myosin interactions it has? Yes, I could potentially manipulate things with contractility. Inotropic, ionotropic drugs, you might read about that. But that has nothing to do with the frequency of contraction. So this action potential, the way the cell is monitoring its movement of ions in and out, has importance on the heart and its ability to, one, you can pull it out of the body and it'll still beat because it's not tied to the nervous system and how it gets told to contract. Second, the heart is always going to have, again, a limit to how many beats it can produce because look how many seconds it takes for one action potential. So remember how there's this whole theory of we have a max heart rate? And usually they say like 220 minus your age, Well, if you do the math, if every contractile cell contraction is 250 milliseconds, how many of those action potentials can you fit in a minute? You're going to have a maximum number of potential heartbeats in a minute that you can produce. All right? And again, that long action potential is going to also influence that you have to give the heart, with this long absolute refractory period, there's always going to be a period of time where the heart is going to be able to relax, stop contracting, and as it stops contracting, it's going to be able to refill with the blood. So when it goes to its next contraction, there's blood inside the chambers to push out. Okay? All right. I'm stopping there because that's a lot to digest. Okay? We'll go through this one more time on Friday, and I'll hit a little bit more on, okay, now we understand why the heart and the nodal cells and the contractile cells, what are the ions coming in and out? Now, how do we manipulate those ions? All right, and so then we'll go into, again, there's some of the things that make the resting membrane potential more of a straight line upwards versus what makes the resting membrane potential flat lines. We'll go through some of, again, why I can manipulate pieces and parts of the ions and the calcium and potassium coming in and out, and how that could potentially make my heart cell give me a higher peak on the contraction or twitching. That's contractility, right? And again, why is that different from my skeletal muscle? Which, remember, if I frequency of contraction, I start to get wave summation or tetany. Okay? Remember that? This does not happen in the heart. Okay? And we'll go through some of, again, these ions and channels on the membrane of the cell. All right, I understand this is hard, this is in the weeds, and this is, oh my God, I don't like electrophysiology thinking right now, and that's okay. So go back again in the cardiac unit folder, go back and watch the resting membrane potential dude's lecture. He'll talk about things on a neuron. Then come back and listen to this again. Come back and again, figure out why the neuron sodium wants to come in, why potassium wants to leave, why calcium wants to come in. All right? Figure out a little bit more on why those ions want to go the direction they want to go. Maybe go back and look at again what is making in skeletal muscle, what makes actin and myosin cross bridge, how that happens with calcium release from the sarcoplasmic reticulum, how we get a muscle cell to increase its force of contraction. Again, you have these comparisons to then, okay, this is how the cardiac cells and the nodal cells work and why they are doing it that way. All right, so I'm going to stop the recording. Questions? Or we all just need time to let this marinate and digest? Okay, Corey, remember that this is one very, very big piece of the cardiovascular system. And if you decide to be a nurse and you decide to be a nurse in a dialysis unit, you decide to be a nurse in the neuro unit, you decide to be a nurse other places, you might not necessarily get to this detail or always be required of this detail. But if you're going to go work on a cardiovascular unit, you're going to work with patients post cardiovasculars, pacemakers, this is going to be what you see. Maybe in the ICU, it's going to make more sense when you start seeing the patients and you start seeing the drugs and you start understanding why the drugs are doing what they're doing. But it's hard right now because you're hearing it for the first time, and we are at a microbiology, very weed in the weeds level. Okay. So if everybody's feeling like Corey right now, that is completely expected. All right. Don't freak out yet. What I want you to take away is if I was to throw up that nodal cell picture and I asked you what is going on at phase 4, you can say phase 4 is the unstable resting membrane potential. Sodium's leaking in, potassium's leaking out. If I ask you what's happening at the 0, that's the rapid depolarization of the nodal cell. That's when calcium's coming in. What's happening at 3? That's the repolarization of the action potential of the nodal cell. That's when potassium's leaving. That's what you need to focus on memorizing, okay? If I show you the big thing of the contractile cell, 4 is the resting membrane potential. And remember, 4 is the unstable pacemaker potential where sodium's leaking in, potassium's leaking out of the nodal cell. If you look at the contractile cell, 4 is still the resting membrane potential, but in a contractile cell, it's a flat line. So what is happening at 4? Nothing. Sodium's kept out, potassium's kept in. Calcium inside the cell is sequestered in the sarcoplasmic reticulum. Nothing is happening. Then phase 0 hits, and it's like a straight line up. Rapid depolarization, action potential initiated. Sodium is coming in. What's happening at the 1? Sodium has stopped coming in. Potassium is starting to go out. What's happening at the 2? Potassium is still going out. Calcium is now coming in, and we've hit a little bit of a plateau. Calcium's coming in because calcium is initiating the sarcoplasmic reticulum to let the rest of the calcium out, and now all that calcium is going to bind to troponin. What's happening at the 3? Potassium is the only thing leaving, and we're resetting and repolarizing. Yes, Dora has a kid. Dora has a job. Dora has her own nursing school classes, and this happens to me all the time. I think there's some expectations from the academic support people that she has to have so many people at meetings and she has to do so many sessions, and the prep work for making sure those sessions are happening and you guys are coming and it's worth your while, so she so people attend. I think it's a lot of work, and right now if you have to jettison something, you're going to jettison the thing that is not going to pay your bills, is not towards your future career, and isn't something you have to do for your kid or your family or your job. So Dora had to make a decision to let go of being a pal because it was just becoming more effort and work, and the expectations she was trying to meet to do it, I personally think, are a little unrealistic. Um, Tutoring, all right. Well, she probably is doing this week. Okay. Um, Tutoring is available, and the tutors, they are not having to have so many people in a session. They are not having to like make every session planned and have slides and have videos and have stuff ready to go for the session. Tutors are just like, I'm here, this is my hour, I'm available, you log in, and then you ask me the question and I try to give you the answer. So tutoring is available, and there are tutors that do that. And the tutors have a little bit more control over. They can say, "I'm only available 3 hours a week. Come meet me." Um, you were supposed to be in specials. You have a special right now. Sorry, he skipped a special. Okay, so Dora, I think, is finishing the week out, and then I guess because of you guys, I think I deleted some of her information from the old announcements. I was trying to clean things up, but I appreciate everything Dora has done, but I totally get it that some of the expectations placed on her to do these sessions are just not feasible with everything else going on. And the tutors, if she was a tutor, it probably would be it would probably work easier. So I did put the link up on there on an announcement about Meg. Meg has been a tutor for a while, and she has helped my students in the past. And through the Merrimack system, they are trying to make you have appointments. You have to make an appointment to see the tutor. I don't know if they have drop-in sessions, but Sally would be the person to contact. And again, that information is there. Now, Forest Park does things differently. In Flow Valley, I have no idea. But Forest Park, remember, they have the bone room, and they have tutoring, and they supposedly have stuff available, but you have to email that S. Rusin or Russo to get information on that. Okay, quiz. I think the quiz is due next Sunday. Let me double check that. I don't think you have a quiz until week 3, and the quiz is going to be over the heart. And this upcoming quiz over the heart is going to be, yeah, the quiz is not until week 3, is going to be open book, open notes. Okay, so there is a quiz week 3, not going to have any respondus requirement. So it's just open book, open notes at your own pace. It is timed. Where are you guys? And it's going to be for week 3, and it's going to be due the Sunday 11:59 of week 3. Okay, but it is going to be potentially anything and everything in chapter 20 related to heart physiology and some heart anatomy. So that's why it's towards the end where you've had 2 weeks to go over hearts. All right. Again, you guys, I understand this is hard, but this is important. This will be one of the only times you ever probably get this information, because the next time you get it will be when you're talking about people who are sick and they're on calcium channel blockers, they're on sodium channel blockers, they're on potassium blockers, they're on drugs to try to increase potassium permeability, to increase sodium permeability, or increase contractility of the heart with calcium. So next time you see this, it's going to be in a drug format that's manipulating these ions, and it's going to be, whoa, 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 why, what? And it'll be, well, if you come back and remember this, this is why we care about calcium to come into the cell or not. That we've never cared about calcium before at an action potential, but now we do. And it's because in the heart, the calcium in the sarcoplasmic reticulum cannot get out unless calcium tells it to get out. And the only way to the calcium to get in the cell is for it to come from outside the cell and release the calcium. Okay, so calcium-induced calcium release. All right. So I know it's difficult. Take some time, read it in the book, listen to this again, and then Friday we'll do it one more time, a little bit quicker, and then we'll go through a little bit more of, now that you understand what ions are moving and why, now we can start to add the manipulations. Okay? Any other questions? Okay. Okay, So 80% of the calcium you need to make a contraction happen. So calcium has to bind to troponin for actin and myosin to interact. 80% of the calcium in cardiac cells is inside the sarcoplasmic reticulum. 20% is outside the cell. So in order to get a muscle of the heart to contract, you need some calcium to come in from outside of the cell. So we can tell the calcium in the sarcoplasmic reticulum to come out, and then all together, the calcium coming from outside the cell and the calcium coming from the sarcoplasmic reticulum, all together, all of that calcium binds to troponin, and you get actin and myosin interactions. Okay? Okay. It depends, Sean. If you're talking about a nodal cell, calcium is going to come into the cell during the rapid depolarization phase of the action potential. The calcium is going to come into contractile cells during the action potential, but during the plateau phase. So both nodal and contractile cells are going to have a point in time where calcium ions come inside. But that point in time is at a different point in the action potential. Nodal cells, it's when the rapid depolarization is happening. Contractile cells, it's during that plateau phase, which is a new thing because we don't have plateaus anywhere else in our action potentials. And the implications of that plateau being there are massive in terms of ensuring contractile cells of the heart always have these long electrical events, and that gives them time to fully contract and relax. So when I ask them to contract again, the heart cell has had some relaxation and had some time to fill with blood. And so anytime I ask the heart to contract, I have a chance that there's blood in the chamber to then squish out. Because it does no good for my heart to contract more forcefully with more oomph if there's no blood inside the chambers to push out. Okay? Yes, it is hard. Let it marinate. Okay? Nodal cell. Okay. Again, the nodal cells, calcium, all right? Both contractiles of the heart have to have a place where calcium is going to come inside, because 20% of the calcium on average for a contraction has to come from calcium that comes from outside the cell. In a nodal cell, the calcium comes in during the rapid depolarization phase of the action potential, the phase 0. In the contractile phase, the calcium comes in from outside the cell during the plateau, the phase 2. Okay? So rapid depolarization of the contractile cell, rapid depolarization of a skeletal muscle cell, rapid depolarization of a neuron, phase 0, is all sodium. Usually when we think of rapid depolarization on a membrane, sodium rushes in. In a nodal cell, phase 0, and this is a big star exception, the rapid depolarization is calcium coming in. And in those, just like in the contractile cells, there has to be a point in time that some calcium comes in, and then that calcium coming in binds to the sarcoplasmic reticulum membrane proteins and releases the rest of the calcium. And then now all of that calcium binds to troponin. Troponin pulls tropomyosin off of actin. Actin and myosin cross bridge. Okay? I know. Let's stop here. I want everybody to have a good lunch. I want everybody to let this go for a little bit and want you to come back, read it, re-listen, and watch it. When someone has high levels of potassium and low levels of calcium, does this? Yes! Oh, John, you're jumping ahead. Remember, people worry about their potassium, their sodium, and their calcium levels. Go back and watch the video for the guy that did this, the neuron. If you screw up what should be normal for extracellular sodium, potassium, chloride, calcium levels versus what is intracellular sodium, potassium, calcium levels, you screw up resting membrane potential. And if you screw up resting membrane potential and cells like skeletal muscles, cells like smooth muscles, cells like neurons start having unstable resting membrane potentials, you start having problems. Okay. Our heart, our nodal cells already have an unstable resting membrane potential. This could potentially, again, make that slope influenced now by factors of what is the concentration of sodium and potassium and calcium and chloride outside the cell versus inside the cell, and again affect that sodium leaking in and that potassium leaking out. John, you are ahead of me. You are getting a little bit ahead, but go back and watch that video. And again, this is why old people are worried about their calcium, their sodium, and their potassium levels. Yes, you're ahead of me. Okay.
I have a quick question.
Yes.
So, for all of this, for like the contractions and everything, is this just for the heart, or does that include any contractions in the body, like whenever you're in labor or something? Like, or is that completely different?
Skeletal muscle, cardiac muscle have striations. They have the sarcomere. They have the overlapping actin and myosin, troponin, tropomyosin, nebulin. Remember that stuff from last semester or your 207? So calcium coming in, calcium binding to troponin, troponin pulling tropomyosin off, actin and myosin cross bridge, ATP is present, myosin pulls, the sarcomere shortens. That is how contraction happens in the heart and in skeletal muscle. The uterus, the smooth muscle around your GI tract, the smooth muscle in your airways, the smooth muscles in your ureters, that is a different mechanism. And if your teacher didn't go over that last semester, calcium is involved, but calcium binds to enzymes, and the big enzyme is calmodulin. And so calmodulin and calcium can make actin and myosin link up. And in smooth muscle, remember, they don't give you 100% contraction and 100% relaxation. They give you a little more contraction, a little more contraction, or a little less. So some of the actin and myosin smooth muscle is always interacting, and calcium and calmodulin, when you want more contraction, make more actin and myosin link ups. And when you want less contraction, you take away calcium and calmodulin, and that makes less actin and myosin, and so your smooth muscle is less contracted. So your answer to your question is it depends. Does that make sense?
Yeah, it does.
Okay. And it's complicated. That's the other answer. Okay. So let's just focus on cardiac. And again, because cardiac and skeletal have those actin and myosin interactions, if you don't remember how sarcomere shorten and some of, again, the importance of calcium and troponin and tropomyosin, that's all in chapter 10. That's all back in the skeletal muscle chapter. Okay. Yes, I know. It's kind of like, oh my God, I actually have to remember all that crap. A little bit, yes.
I had a quick question, Dr. G.
Yes. Who am I talking to?
This is John.
Okay, John. Who's getting ahead of this?
The contractile cells, are those the only one with voltage-gated channels to allow the... No?
No, I mean, voltage-gated channels are any place you have an action potential, because remember, action potentials look always exactly alike, and that's usually because there's voltage-gated channels involved. All right. So contractile cells have voltage-gated sodium channels for their rapid depolarization phase 0. They have potassium, voltage-gated potassium channels that are going to be involved in the initial repolarization, and then eventually all through the plateau and all through the repolarization. And then they have those voltage-gated calcium channels during the plateau phase. Okay. Your nodal cells have voltage-gated calcium channels that open when the resting membrane potential that's not stable hits threshold.
Okay, right? To cause repolarization?
Yeah, and then it has its voltage-gated potassium channels to, again, repolarize after all that calcium's coming in. Skeletal muscle cells have voltage-gated channels right at the motor end plate where the acetylcholine receptors are. Those are ligand-gated channels for sodium entry, but when enough sodium comes in there, that'll trigger voltage-gated sodium channels to open, and that's what is the electrical spread of energy across the membrane and into the T-tubules. And then there's voltage-gated channels on your neurons. Again, anywhere you have an action potential, there's voltage-gated neurons at those nodes of Ravanier where sodium is going to rapidly enter and then potassium leaves to do the repolarization. Okay? And it gets more complicated, John, because when you eventually get to it, some of your voltage-gated channels are going to be proteins that they're going to call fast gates. Some of them are going to be called slow gates. So it gets more complicated. At every one of those proteins, whether they're fast gate, they're slow gate, they're voltage, they're ligand, they're all drug targets. So when eventually you get to pharmacology, that's why there's not one drug to treat the condition. There's many drugs because every protein involved in every piece of this puzzle could potentially be a drug target.
Okay, so if you had something like if your heart's beating slow and you gave like atropine or something, does that
Okay, I'm not
bind
going
to
to ask
the...
you that because we're not going to talk about drugs. The only
Okay.
thing I would say is, like, which of the following might be a way to get a resting membrane potential of a nodal cell to become more tachycardic, to increase its rate. Sympathetic output, caffeine. I'll mention, like, because caffeine does it as well. And then some, like, I'm not going to be into the weeds of different drugs. I usually will hit one or two things. So on the flip side, what's a way to get the heart rate and the resting membrane potential to be less likely to hit threshold? That would be parasympathetic, and that's what we're going to do Friday. I'm not going to try to hit all the potential ways to get sodium in during the resting membrane potential and all the ways to get it out, because that's, again, you got to build on this. That's your next class and your next class. We just need you to get the basics right here, right now. But I'm glad you're thinking that way, but I don't need you to think that way yet. That's next class.
Okay, thank you.
Yeah. Now your books may have a few, like those book questions or those book examples. They may hit on a few examples that I don't. I'm always going to probably err on the more cautious side because, again, I don't want to get too much into what are the drugs, what are the signals, what are the different things, because that, again, that's a whole other semester we could spend on just that. Okay. Any other questions from you guys still around? Okay, I guess my son skipped his PE or his library, so I need to go yell at him. And I'll try to get this video recording when it pops up. I'll try to get it posted on YouTube for later. But you guys take a break. This is really, it's hard on your brain. Take a break, eat lunch, and either wait till tomorrow to go over it or, do it later tonight, review it, and then we'll go again over it on Friday. Okay, we'll see again on Friday. All right, I'm going to say goodbye. Thank you all for putting up with me in my stairwell of my basement. Bye, guys.